I am a geek for viruses. I love viruses. It's one of my passions in life. We tend to anthropomorphize viruses. The virus wanted this, the virus wanted that, but viruses aren't alive. A virus is a machine that makes more of itself. By accident, it makes itself better occasionally, but for the most part, it makes itself worse.
Welcome to Unplucked, the official podcast of the Poultry Science Association. This is the podcast where we strip away the fluff and get real about poultry science from bench to barn. I'm your host, Andy Vance, Executive Director of the Poultry Science Association. In this episode, we sit down with Scott Kenney, a professor at The Ohio State University, to unpack the science behind avian influenza's surprising appearance in dairy cattle and what it means for poultry producers. From viral receptors to cross-species transmission, Dr. Kenney breaks down the virology, the risks, and the urgent questions the industry needs to ask now as we move forward in tackling this disease outbreak. His areas of research interest include cross-species transmission of positive strand RNA viruses and development of viral infectious clones and reverse genetic systems. He studied viruses including hepatitis E, porcine delta coronavirus, African swine fever, and turkey reovirus. So a broad spectrum of zoonotic diseases in a variety of different species with implications for the food animal industry. Dr. Kenney, thanks for joining us. I want to again mention that you were one of our speakers during our scientific program at the PSA annual meeting in Raleigh, and appreciated you sharing your expertise. Obviously, high-path AI is maybe the most pertinent animal health topic we're dealing with in the poultry space. Your research, as I mentioned, spans multiple RNA viruses and host species. Would you walk us through your background in zoonotic and cross-species viral transmission and how you got into this area of research?
Sure. So first of all, thank you for having me, Andy. It's a pleasure to be here, and it was a pleasure to be invited to the Poultry Science Association to give a talk on influenza. So I am a geek for viruses. I love viruses. It's one of my passions in life. A lot of people are like, I like fantasy football. I like watching football. I like to play with viruses and understand them and how they work. So might be a little bit strange for some people, but definitely one of my favorite things in life to do. I had a lot of really good high school teachers, and people are amazed when you're like, wow, a high school teacher had that much of an effect on your life. And I'm like, yeah, well, AP biology, we had a great teacher who, introduced me to genetics and those types of things. And I've always been fascinated. I was not the best high school advanced placement biology student, but I loved it so much that I was fascinated by it, and I've continued on over the years. I originally wanted to be a veterinarian, realized that maybe that wasn't the best career choice for me towards my senior year of college. And I sat down with a career counselor at Penn State, and he said, what are your passions? And I said, well, I've had a lot of great microbiology classes. I kind of want to continue on in that area. And he's like, well, there's this thing called graduate school, and they'll pay you to go there. They'll give you a stipend. I was like, I can't say no to that. This sounds great. I can do what I love, and I can get paid to go to college to learn more about it. And I got to do a lot of human-based research using a poultry model system. So we use something called Rous sarcoma virus, which is a simple retrovirus that causes bone cancer in birds, basically, but it does similar things to human immunodeficiency virus. And so for my graduate work, I actually got to understand how human immunodeficiency virus worked by working with Rous sarcoma virus, which was this poultry virus that didn't infect humans, so it was safer to use than using HIV. From there, I did my postdoc, as you mentioned, at Virginia Tech with XJ Mang. His lab worked on mostly hepatitis E virus, and hepatitis E virus also has an avian strain of hepatitis E, which also doesn't infect humans, so it makes it safer for us to work with. And so we used a poultry model in XJ's lab, but hepatitis E also branches out. So it also infects a number of different mammalian species, including rabbits and pigs and, all kinds of agricultural species. So that sort of set me on my way to looking at more general cross-species viral infections. I also did a lot of vaccine research for things like porcine reproductive respiratory syndrome virus, porcine circovirus. And so it was a mind-opening experience going for a number of different viruses, but still retaining that passion for crossover potential of animal viruses that can infect humans. And it's been a good fit. So I joined Ohio State in 2016. The one neat thing that we have is this biosafety level 3 facility. And it's one of less than maybe a dozen facilities in the country where you can do high containment pathogen research in large livestock species. So we can do high-path influenza, African swine fever, Japanese encephalitis virus, SARS-2. We can do a lot of high-containment research using this facility and actually put it into the host animals. And that's given me, a great advantage of being able to look at pathology and disease occurrences in these animal hosts themselves, whereas a lot of institutions in the United States can't do that type of research. So I think that's kind of where my passion lies, is doing these animal model systems and disease pathology and looking at transmission. And I'm lucky enough that I can do it both in the laboratory setting where I can, manipulate the virus safely, but I can also take it into the host species and, infect that animal also in a safe manner. And so I think that gives me a very unique perspective from lab to bench to actually in the animal host, and putting all those factors together to understand how those viruses work and transmit and cause disease.
Well, and I suppose I should admit my pro-Ohio State bias here as, an alumnus of the College of Food, Ag, and Environmental Sciences and, current PhD student there, though not in your area of expertise by any stretch. I'm one of those wacky would-be social scientists. But I want to say the work you're doing there, Linda Safe as well, Ohio State is really, in your Center for Food Animal Health has really developed, I think, international renown in this area of looking at zoonotic viruses and diseases more generally. These cross-species transmission has really become, one of the foremost facilities in the world dealing with these questions, related diseases. Your lab in particular, as you've described it, you're exploring these host virus interactions with pigs. We're now talking with poultry species into dairy cattle. When you approach a virus like H5N1, particularly in a scenario, now where we're being expected to look at these unexpected species such as dairy cattle becoming vectors, how do you approach that? So when you started looking at this particular strain and this particular outbreak, what's the process that you employ to try and understand what's going on in the birds, in the cattle, in the people potentially, and how we understand the transmission across those different vectors?
Yeah, so that's a great question. So we were actually very lucky that influenza virus, high-path avian influenza, is actually called a select agent, which means it's on a list of government-specific pathogens that are highly regulated in their ability to work with them. On our campus, we previously had a researcher named Changwon Lee who was doing avian influenza research, and I took over his select agent clearances when he moved back to the USDA. And so we actually had a leg up that we had that clearance in place. So as soon as the virus outbreak occurred, we're also lucky that I would say lucky, but we had one single outbreak that occurred in Ohio, out in northwestern Ohio. And some of our faculty members from the College of Veterinary Medicine, along with the Department of Agriculture, they were on the ground when that outbreak happened. And so they managed to take samples. They sent some of those first samples to Richard Webby's lab in St. Jude down in Tennessee. He sequenced it and he was allowed to send it back to us. And we were allowed to start working on it almost immediately. Originally, it started out as an unknown outbreak. No one really understood what was happening in these dairy cows. And when people were saying, it kind of mimics influenza, people were like, oh, that's crazy. Cows don't get influenza. Until someone actually ran that first PCR and they're like, wow, this is influenza in cattle. We've never seen this before. You really don't know what's going to happen or how that outbreak is going to unfold. And so you need to have the basic principles of both epidemiology. So how that virus is spreading, within and around a herd, how it's replicating in that animal, what kind of disease it's causing, and what kind of routes it's using to get into those animals. And so because we have this large animal facility, we were actually able to get full-size dairy cows. So we're talking, 1,500-pound dairy cows coming into our facility, and we were able to challenge them. Typically, you think of influenza as a respiratory pathogen. So obviously, you want to start with putting it in their nose and see if they get sick. But from the epidemiology in the field, we knew that it was affecting, milk production. So are these animals getting it nasally and then it's going systemic and affecting the milk production? Or is it some other manner of transmission that's causing it to affect, milk production? And so to answer those questions, you really need to experimentally challenge those animals. And so that's where we started. We got that virus from Richard Webby's lab. We grew it a few times in a cell culture. And so we have cells in the lab that we can replicate the virus to make a large stock of it. And then we can take that and actually put it into these cows. And like I said, PAR facility is spectacular because it's high biocontainment, very secure. None of that virus is ever going to get back out into the environment. So we can have it and we can monitor very closely these cows. And some of our first studies, we took it, we put a high dose. So I think we said like 10 to the ninth, I think was the titer. I'd have to write it down to tell you what the exact amount is. I'm a biologist, not a mathematician, so it's okay.
I always joked I was a comms major because I was bad at math and science, so I empathize.
Yeah. So we took this high titer virus and we basically used what's called an atomizer. So it's like a syringe with a little disc and it causes nasal mists. And we sprayed that into these cows' nose. And then we watched them for, 15 to 18 days to see how they would get sick, what would happen. At the time, it was March of 2024. And we had some other incidences out of Michigan where they were saying that there were some spillover events into chickens or poultry. And so I said, well, maybe in our facility, we should also put some poultry to see whether or not, it transmits between the poultry and the dairy cattle. So we inoculated these cattle intranasally. To our surprise, they didn't get very sick. We were expecting much more severe disease based on what was being seen in the field, where cows were spiking really high fevers and, milk was dropping off to almost zero production. But surprisingly, when we gave it intranasally, the cows were, for the most part, fairly normal. All of our experiments are terminal, so these cows are euthanized, and we, dissect them to see where the virus is and what kind of pathology, so what kind of disease these cows are getting. And really, we only saw some hemorrhaging, like right in the nasal passages where we had given the initial inoculum. So we had a little bit of infection, a little bit down the trachea, and maybe a little bit in the lymph nodes. But to us, it hadn't gone systemic, never reached the mammary glands, so it seemed to be fairly restricted to the upper airways. And to our surprise, like all 8 of the chickens that we put in open air caging, they all survived. So if, talk to anyone in the HPAI field, you're like, oh, you give them, influenza, high path influenza, those chicken are gone in 3 or 4 days. That's just the way it works. So to us, that indicated that the virus was going into the cow's nasal passageways, but not really coming out very efficiently. And I think, that also speaks to the engineering controls in our facility. So the PAR facility is designed to prevent exposure to the people in the facility to the pathogens.
Sure.
And so I think a lot of those engineering controls took care of any of the little bit of virus that came back out of those cows intranasally, that were intranasally inoculated. This was the first time we ever worked on dairy cattle in the PAR facility. So it was really a learning experience for us as scientists. And as I mentioned, those 1,500-pound cows, they kind of go and do what they want when they want. So for us, it was a learning experience how to manipulate these cows in high containment while maintaining safety and, all those biosafety and biocontainment protocols. So yeah, our first experiment, a little disappointing. We didn't see any severe disease. So we next moved on to intramammary inoculation. So we took the virus, fairly similar high dose, put it directly into the mammary glands. So you can use a cannula and just squirt it up there. And those cows spiked 106.5-degree fever in 24 hours and had neurological issues. And we had to euthanize them within 2 days. So
Wow.
For us, it was very telling that direct exposure of the virus into the mammary glands is probably the most likely route to severe disease. And so we learned a good bit about that. By this point, we hadn't put any more chickens back in the facility. I kind of wish we had maintained them a little bit longer.
Yeah.
And we really haven't done a lot more with chickens because our funding is mainly through the SEER network. And it's mainly to look at dairy cattle specific and not necessarily the poultry host. So really, we did the intramammary, and then we actually did a titration. So we took different amounts of the virus and put it into each mammary quarter. So if you know a cow, they have 4 teats, and each one is an individual quarter.
Right.
And the virus doesn't spread outside of the individual quarters. So you can inoculate each individual quarter, and you can use it as a separate experimental unit. And so what we did is we put 10 to the 3, 10 to the 2, 10 to the 1 infectious units of virus into each individual teat. And it was very interesting that the amount of virus that comes out is like astronomically high. Like you get upwards of 10 to the 11th or 10 to the 12th infectious units of virus coming out of an infected mammary gland. And we saw that all the way down to 10 particles. So you can put 10 individual virus particles in a cow's teat, and within 24 hours, you're getting back, 10 to the 11th infectious units, which it's amazing.
Regardless of what the volume was that you put in, they were multiplying that significantly.
Yeah, so the mammary tissue of cattle is like an extraordinary virus reactor to make new virus particles, and fairly susceptible because you can put in a low dose. So if you look at a lot of the research with poultry, they say that the infectious dose is around 10 to the 3, so around 100 infectious particles necessary to make a sustained infection, whereas at least some of our data and limited numbers suggest that only 10 particles. So you're talking tenfold difference in the infectious susceptibility in that tissue.
And very different from what you saw with the intranasal inoculation.
Yeah, exactly. So like I said, the intranasal, the cows were mostly normal and healthy, and intramammary, the cows got extremely, extremely sick. And so for us, there's no smoking gun, right? We don't have how are those cows getting sick? How are they transmitting between farms in the wild? And we don't have necessarily the best answer for that. What we would say is because of the low susceptibility of the mammary tissue and the high amount of virus that's secreted in the milk,
Mm.
that there's probably a lot of different ways that that virus can be transmitting. And it's probably centered around the milking parlors because you're milking hundreds to potentially thousands of cows a day, and you're making like a massive amount of virus. So any spill of milk, anytime you're stripping those teats of the cow to initiate milking, you're just putting virus basically on the floor. You're making aerosols. So basically you're just creating this environment where there's a whole bunch of virus potential that can then be either manually inoculated into a teat or, subclinically inhaled into the cow's respiratory tract and maybe transmit it that way asymptomatically. Again, we still don't fully understand the story. There's a lot of good epidemiological evidence that suggests that the milking parlor and that contaminated milk is probably one of the major contributors of spread of that virus.
Well, thinking, in that context and looking at, work you've done in the past with some of the porcine diseases, Delta coronavirus, and others that you've worked on, what are some of the key virological factors that would enable a virus like HPAI to jump from birds to cattle, cattle to birds, and so on? What do we know about virus transmission, viral behavior generally that might inform either future research on the subject and some of the management decisions we might make in response during this outbreak?
I don't do a lot of comparisons between virus species because each one has its own little nuances.
Sure.
A lot of them have their niche factors, but in general, viruses have a higher fitness if they can infect a multitude of hosts. So we tend to anthropomorphize viruses. The virus wanted this, the virus wanted that.
Right.
But viruses aren't alive. They basically function based on the principles of evolution and transmission, right? So virus makes a whole bunch of itself. Some of that is better than the stuff that went into the animal, and that stuff that's better survives and transmits to the next animal. So really, you have to think of it as a machine. Like a virus is a machine that makes more of itself. And so by accident, it makes itself better occasionally, but for the most part, it makes itself worse. So a lot of those mutations are deleterious and kills the virus, but that one single mutation is better, gets into another animal, and then it replicates even more effectively. With influenza, there's a lot of different factors that allow it to spread and become more efficient. Receptor binding specificity, that's a lot of what we talk about when we do influenza virus. So influenza has, hemagglutinin protein and a neuraminidase protein on its surface. And that hemagglutinin is what binds to the sialic acid. So sialic acid is a sugar moiety that's on the outside of cells of the host. So in the upper respiratory and lower respiratory tract, these lung cells or these tracheal cells have these sugar moieties on them. And so with influenza, birds typically have something called alpha 2,3 linked sialic acids, and mammals, including cows, predominantly have alpha 2,6 linked sialic acids. And you're really going into the weeds in chemistry of alpha 2,3 versus alpha 2,6, just happens to be where certain chemicals are linked on these different sugars. But what happens is this can link, in birds, it likes this alpha 2,3. When it tries to adapt to mammals, it's looking for a mutation in HA that can allow it to bind to alpha 2,6. And then you have those lucky viruses that like to bind to both alpha 2,3 and alpha 2,6. And those are the scary ones because it can sort of go back and forth between mammals and birds. And the other thing about influenza viruses is they love to recombine. So unlike the other viruses I work with, Delta corona and hepatitis E virus, they're both RNA viruses, but they only have a single genomic segment. So they're single genomes, right? They're one long piece of nucleic acid, whereas influenza viruses are composed of up to 8 separate nucleic acid strands. And so each one of these encodes for a number of proteins that make new viruses. And so what happens with influenza is if you get 2 different strains of influenza in the same cell, they will reassort these 8 fragments, and what comes out is completely different sometimes than what went in. And so it's taking different pieces from different viruses, and sometimes these are functional better than other viruses that went in originally. Hepatitis E virus, we typically don't see a lot of recombination, but the coronaviruses, they do recombination. So they'll take that single strand of nucleic acid, and at certain points they will, if they're co-infected with another closely related coronavirus, they'll chop their genome and the genomes will recombine into another solid genome. But that's obviously less efficient than if you have 8 strands of nucleic acid that are just mixing and matching between the 2 virus strands. So Danger of influenza is that one, it can infect multiple hosts using sialic acid because it's a fairly broad and available receptor, but then it can also reassort very readily. So it likes to take genome segments from different viruses and mix and match. And what comes out might be even scarier than what went in. And then finally, you have things like the hemagglutinin cleavage site. So hemagglutinin, as I mentioned, that's your receptor for the virus to the host cell. And so you have two different kinds. With high path avian influenza, the amino acids form this thing called a polybasic cleavage site. And in low path viruses, it's a monobasic site. And that really has to do with the host enzymes that can cleave this receptor protein. And that receptor cleavage is necessary for viral binding and entry into those cells. Because of the polybasic cleavage site, the viral entry is much more easily accessible in both the upper and lower respiratory tract of the host, whereas the monobasic is mostly in the upper respiratory tract. And that's really what leads to that severe disease. So you're looking for a lot of different things that could potentially lead to better viruses that arise.
I want to shift from transmission and virology to risk and monitoring. I'm thinking back to when you were talking about your PAR facility and biosecurity and kind of managing these critters, both mammalian, avian, as well as the viral. How concerned do you think a poultry producer, if I'm a producer listening to this, a facility manager or somebody who's involved in the live production side, about high path AI entering my flock from some indirect route from an infected cattle operation, for example, based on what we've learned or studies that you've done thus far? How concerned should I be about that if I'm somebody on the live production side, that these dang dairy cattle are about to make my life even more difficult from a high path transmission standpoint?
So again, the high path is always what keeps poultry and poultry farmers up at night because it's such a lethal disease. And so they don't necessarily need more to worry about. Luckily, if you look at the USDA website that reports dairy cattle outbreaks and new dairy cattle outbreaks, there's only been a single new case reported, and that was like August 1st. And so it looks like a good sign that we've potentially run the course of this initial outbreak and that it's petering out in California and hopefully it doesn't come back around. I mean, we always have to be vigilant that, there's not a new introduction during the migratory season, which is, coming up very shortly in the next few months, when those migratory waterfowl will again cross the United States and potentially deposit more influenza virus non-discriminately everywhere. But yeah, so biosecurity is probably one of our best methods to keep that virus out of the flocks. Anytime you have a virus factory, like undocumented dairy herd that may be asymptomatic and they're pumping out millions of or billions of virus particles into a waste lagoon that's not treated, that definitely becomes a very scary possible transmission source, because those lagoons, they're kind of like a oasis in the middle of a desert. So all of these other animals will come to that lagoon to seek water. And if you're putting untreated milk into that lagoon, you have the potential for huge amounts of virus to be going in there. And so it's very important that we monitor those production facilities and try to mitigate that amount of wastewater that might be infected or waste material or milk that's deposited into that lagoon. Very good biosecurity tip would be, even, if you have that milk, you want to pasteurize it, because pasteurization will kill influenza virus. So even that waste milk, you should probably consider either treating with some kind of detergent or pasteurizing before you're releasing it to the waste lagoon. And that's part of where surveillance comes in. So I know it's expensive to do that pasteurization sometimes, but if you suspect any kind of biohazard that might be in your milk, always a good idea to treat it rather than release it back into a cycle and have more virus particle that can then, get into more waterfowl, get into, mesocarnivores like mice or skunks or, any of those animals that would visit that water hole and then potentially come into close proximity to neighboring farms, including poultry facilities. So anytime you can mitigate a potential transmission source of virus, it's highly recommended. And for what we know, poultry farms and swine farms, because of the threat of influenza, that's part of the reason we have such high biosecurity on those farms, because we know that influenza is, highly transmissible and it can infect those species and cause significant losses. The dairy cow thing is new, and so their biosecurity isn't quite on the level of poultry and swine. That's something that maybe we look into to prevent future outbreaks, is to help those dairy farmers and those, cattle farmers maybe improve those biosecurity and, limit people coming into the milking parlor and leaving the milking parlor. And there's ways that it will eventually help to break, those kind of transmission events.
Thinking about our blind spots, potential gaps in biosecurity. You've just given us some good examples there. Thinking about any other mutations, recombinations, you talked about how the virus likes to make more copies of itself and it will adapt or evolve, if you will, with that goal in mind. Do you see any signs or any warnings that this cattle associated strain could adapt further in poultry populations? Is that future research that we need to conduct yet, or what's on your radar in that regard?
Really, this strain is an avian adapted strain. And so it's pretty much, I wouldn't say perfect, but it's already well adapted to migratory waterfowl. And so migratory waterfowl are fairly close to poultry, or they are sort of poultry, but the virus is pretty optimal for infecting chickens already. And so, from my understanding of the literature, there hasn't been any reported amino acid mutations that appear that the virus is further adapting to the poultry farms that it's infecting, or even in the dairy cattle itself. More worrisome is, that adaptation away from poultry and into mammalian species. Because if it becomes extremely efficient at mammalian species, then you have something that not only can infect dairy cattle, but it more efficiently affects humans. And so eventually you get something that can cause a pandemic spread of avian influenza in humans. And that would probably make something like SARS-2 look like a cakewalk at this point. So something we definitely want to stay away from is that mammalian adaptation. I'm concerned about poultry, but those viruses are going to kill poultry the way they are right now. And we want to avoid that mammalian adaptation from a human perspective. So if we wipe out all the humans, we don't necessarily have to worry too much about the poultry health.
Well, and you made a good point there. We kind of already have an idea of the worst case scenario, so to speak, on the avian side of things, because we've been dealing with the massive depopulation and so on that's come from that, given the virulence of that particular strain. But I'm glad you kind of gave a good segue into this concept of One Health. So, with a One Health perspective in mind, what are the lessons that both poultry and dairy industries need to take from this? You've talked a little bit about the biosecurity, but to try to ward off that potential for a pandemic that I think we're all at least a little concerned about, what do we need to take away from what we've learned so far about the interconnectedness of animal and human health to be in a good position to ward off and or respond to what might come next?
Influenza is definitely a One Health issue. It infects quite a number of species, both domestic and peri-domestic, and humans. And so it's critical that we mitigate this virus spread in these domestic animals, because eventually what happens is it does continue to mutate and become problematic for these other species. And so I have a t-shirt that says, "What doesn't kill you mutates and tries again." And that's very true with influenza virus. Like, it is actively evolving in the host species. Now that it's E313 and D1.1 seems to have gained a small foothold into mammalian hosts, we worry that it continues to evolve silently or not completely undetected. But once it hits on those 4 or 5 mutations in its amino acids that allows efficient replication in humans, the genie's out of the bag and we're in trouble. So again, monitoring the situation, if we can use vaccines to help mitigate the spread of this virus, I think it's going to be an important aspect, veterinary virology aspect, to help prevent spread in the large domestic populations, which are serving as this bridge between humans. Because we have this very important population of humans who are interacting daily with these animals, and they're serving as sort of that bridge area between taking direct interaction with animals and those animal pathogens, and then taking that back to the general human population. And like I said, you're setting up for animal to human, and then again to the second human, which then allows the virus to passage a couple times. And eventually, like I said, that evolution favors the virus finding a new host that's highly populated and ripping through that population. So a lot of concerns out there, and we cross our fingers that our current therapeutics, those drugs and stuff that we can use, the yearly influenza vaccine, we hope all those strategies will protect us with this potential new outbreak. And so typically when we say influenza every year with humans, we're thinking H1N1. And so we hope that that N1 component of our H1N1 flu vaccine provides some cross protectivity in case an H5N1 outbreak occurs, and it doesn't look like it's entering into a completely naive population. And I think some of the worst case scenarios when we've seen that has been like that 1918 Spanish influenza, where you had that complete jump in H and N. that resulted in a huge population loss in humans. So we've evolved since 1918. We have antivirals, we have vaccines. It takes a little while to get an optimized vaccine. So if there's an H5N1 outbreak in humans that is sustained, it takes, quite a few months to get that new H5 variant into a vaccine formulation. So there's that critical amount of time where that virus could be spreading unchecked, causing a lot of damage.
Dr. Scott Kenney, I appreciate you sharing your expertise with us both at our annual meeting this summer in Raleigh and during this episode of the podcast. Friends, I would encourage you, if you want to listen to some more conversations about the evolving H5N1 high-path AI situation, and particularly some of the cross-species transmission of the dairy space, we've had several episodes of the Unplucked podcast that have covered this topic. I would encourage you to go back and listen to those. Some really great conversations from a variety of different perspectives throughout the poultry and dairy industries, the research and veterinary communities, production, and all of the things under the hood going on here as the industry continues to navigate and respond to this animal health challenge. Also, I would encourage you, if you have not subscribed to the podcast, go ahead and do that and give us a rating if you don't mind, wherever you listen. It does help others who might be interested find the podcast. So subscribe and rate if you get a moment. We will be back next week. Would you do me another favor? We are coming up under 60 days now to the inaugural Poultry Science Association Pacific Rim Scientific Conference in Macau, China. If you haven't registered, you still have time to do so and book your hotel at the Grand Londoner in Macau. Fantastic facility. We have a robust scientific program with nearly 300 individual presentations between our symposia, abstracts, poster presentations, really tremendous gathering of researchers from around the world. I think at last check there were 29 different countries represented on the scientific program and among the registrants. So truly an international event coming up this October. You can learn more and register at poultryscience.org. Just click the Meetings and Events tab and look for the Pacific Rim Scientific Conference. Until next week for the Poultry Science Association, I'm Andy Vance.